So we do believe that air is involved and associates with this complex that's quite well known in a couple of contexts. So one context is in the DNA damage response, where this complex is sort of at the balance between the decision to go to death versus DNA repair. I should have mentioned parenthetically that another function of air is that it causes the death of these medullary epithelial cells within 4 days of being turned on. And then associated with repair, there is a transcriptional program and bringing in of chromatin remodeling enzymes. But I guess many of you probably know that this complex here is also involved in RNA transcription, and there are more and more contexts where that's being shown. It's what's called the eviction complex. As you know, in front of the polymerase, you need to sort of move the nucleosome or disrupt the nucleosome so that the polymerase can be effective. So this eviction complex seems to ride along with the polymerase, strip off an H2H2B dimer, often substituting H2AX for the H2A, and then reassembling it back behind the polymerase. So that made us wonder then whether air might not be involved in transcriptional elongation. Before going into a set of studies that would take us a while to set up, we thought we could maybe make use of some data that we already had in the lab. That was our microarray profiling, which we have a large number of subsets in different contexts where we have air or we don't have air. And just to go over this very briefly, as most of you know, on the old Affymetrix microarrays, the probes, each gene has, say, something like 3 to 5 probes, and they're skewed towards the 3' end of the gene. And when you put in your values into the computer, what comes out is basically some kind of weighted average. Now, with the Affymetrix ST1 chips, it's arranged in a different way, where there are 29,000 genes that are looked at, and for each gene, there are about 26 features or probes, and these are distributed all along the gene such that each exon has a few probes that go along with it. And again, if you put your data into the computer, what you get is a weighted average of this whole thing. But what you can do instead is to look at this feature by feature, so exon by exon, using or adapting programs which were devised to look at differential splicing. So we've done that, and just to be very clear before we go ahead, what you're going to be looking at in the next slide is what we do first is take this value of the whole gene, which is air regulated, and then we walk across and look at exon by exon and match how that's differentially expressed in the presence of air versus the whole average of the gene. And so if we do that and take all of the air-induced genes, basically the air signature, what we find is that there's a skewing in the first couple of exons such that their differential expression is different from the average of the whole gene. And that's not true if you look in orange for air-repressed genes or when you look at other transcription factors, for example, KLF. So then what we did was to take these ones which are up here, gene by gene, all of them, I don't remember how many there were, maybe 100 or something, and just walk across the gene probe by probe, comparing the expression of that probe in the air knockout versus the air wild type. And where it's the same, it would be along this axis here, no difference between air wild type and air knockout. And where air induces the expression, it'll fall below here because it's lower in the knockout than the wild type. And there's a very striking pattern that until you get up to about 200 nucleotides, the expression of these individual probes in the absence and in the presence of air is exactly the same. However, once you get beyond about 200 nucleotides, then we know these are air-induced genes, so that that falls very rapidly, and this continues throughout the gene. So of course, we have controls of other genes which are not air-regulated. And I just want to state very clearly what we think that then is that on air-induced genes, the polymerase is already loaded, but it seems to be in a stalled or poised state, and that air acts by lifting this stalling. So I'd just like to end by showing you a model of one part of what air does, and it is a model, and we know that there are additional experiments that we can do to confirm this model, but we do think it's correct. So both from our lab's data and the data from the Pedersen lab, we do not think that air takes a gene that's totally turned off and turn it on. Instead, it takes a gene that's being expressed at low levels, has a little bit of polymerase, and upregulates expression. So we think there are at least two things involved in the targeting. One is hypomethylation of H3, the terminal H3 tail, as I mentioned, H3K4. And the second thing is that it will be in those regions where it can interact with topo and polymerase. This interaction leads to double-stranded break. This calls in other transcription factors and remodeling factors, and eventually leads to elongation. From studies that I haven't talked about, we know that this also is accompanied by methylation now of the H3K4 residues, as well as acetylation of this H3 tails. Now, this H3K4 methylation comes from the result where, as I told you, if you take a cell that doesn't have air expressed and look at the potential air-induced genes, they're in hypomethylated regions. However, if you then transfect air and look at the air-induced genes, they're now H3K4 trimethylated and dimethylated. And then as far as acetylation, our information of that is coming from the whole set of other studies, which I didn't get to talk about today, but just to give you a tantalizer, we know that one of the genes which is required for air induction of genes, and actually specifically for this lifting of the pause, is MIST3, which is histone acetyltransferase. So we do think that that's somehow involved in this. And then just to just put it in the whole context, of course, I only told you about two sets of interaction. There's also the whole world of air's involvement in controlling pre-messenger RNA splicing. This seems like it does a lot of things, but we think about that in the context of the building sets of data that gene expression is no longer thought of as something which is a linear transcription followed by splicing, followed by export, but that all of these things are happening simultaneously. And there are several examples. If you mutate a splicing factor, that will have an effect on transcription, or if you mutate a transcription factor, that can have an effect on splicing. So that's my story today.